Hi everyone, welcome to the next video. So today what we'll try and do, and with the fixation probabilities that we discussed in the last video, when a mutation's fate is being decided purely under the action of drift, or when it is being decided under the action of drift and selection put together, we derived these relationships for different probabilities. And today we'll discuss two different regimes under which mutations can happen, and we'll try to get a sense of what is actually going on in a flask or in a chemostat when a microbial population is evolving. So if we go back to the chemostat example, we have a genetically identical population. Let's represent that by these black circles, and in this population we have a blue mutant individual which comes up in the population. And what we've been deciding, what we've been deriving so far tells us that the fixation probability of this mutant, if this is a neutral variant, the fixation probability is simply equal to, so probability that blue individual reaches fixation is simply equal to 1 upon n, where n is the population size. Alternatively, the other case we discussed, so in the neutral case, since the fitness of the blue individual is exactly the same as the fitness of the black individual, selection is not doing anything, selection has nothing to distinguish between blue and black individuals, and it is only chance or drift which is deciding the fate. On the other hand, if this mutation is beneficial, so if blue mutation is not neutral but is in fact beneficial, then its fate is defined by the combined action of selection and drift. And what we saw in that case is the probability that it reaches fixation in this case is simply approximately equal to S, where S is the selection coefficient associated with the mutation that has occurred. And the precise definition of this was fitness minus fitness upon fitness. Let me just do this. So the precise definition of selection coefficient can be represented like this, that fitness of the selection coefficient associated with this particular mutant, in case it is a beneficial mutation, is simply equal to the fitness of the blue individual minus the fitness of the ancestral population. So numerator here represents the benefit that the mutation confers, and denominator is simply the fitness of the ancestor. So selection coefficient is the fractional advantage that a mutation confers to a population. So we did these two cases. So now we know that fates of the populations are going to be decided via these two fates. Of course, there is the third possibility that mutation can be deleterious, but that is a case that we need not worry about because selection will just eliminate it. One way to think about the effect of a deleterious mutation could be that if even if a deleterious mutation is able to survive the action of drift and increase to higher frequencies, then selection will start acting and selection will try to get rid of those variants because they are lower in fitness. So even if they were to escape the action of drift and not be eliminated early on, and their numbers increase in frequency, even then selection will not let them rise too much in the frequency, too much in the population. All right. So given these, now let's take a look at how are mutations actually happening in a chemostat or a flask. And we can think of this as two different possibilities that could be going on inside a flask. Let's discuss these possibilities. So we are going to use two representations, and now in 3 or 4 videos time we are going to move towards experiments, and the rest of the course is going to be just discussing evolution experiments and the lesson we learned. And these two representations are useful in the context of, these are also the representations that we will see in papers when we are discussing results from experimental evolution. So in the first one, let us say x axis represents fitness and y axis represents numbers of individuals. So let us imagine a population in a chemostat which at t equal to 0, all individuals are identical, all n individuals, population size is n, and the fitness of each individual is f naught. So if I were to represent this population in a graph like this, I would identify the point f naught here, and the total population size in this chemostat is n, and hence the state of the population at this particular moment looks like this, that there are n individuals with fitness f naught, and there are no other individuals with any other fitness level, because this is an isogenic population. And the chemostat will continue to operate like this while divisions are happening, cells are dividing, newer individuals with f naught are coming in, some individuals are being washed away, and so on and so forth, until a beneficial variant arises whose fitness is no longer f naught, but it is f naught plus s. So this mutant is carrying a beneficial mutation which has increased its frequency by some amount s. At that point, when this mutation has happened, then this is no longer an appropriate representation of what is happening in the chemostat, and hence we need the representation in that case will change to at f naught and at f naught plus s. Let us say this is n. Now this is represented by the following, that there are n minus 1 individuals of fitness f naught and there is 1 individual of fitness f naught plus s. So this is now this graph is now going to represent the state of the system as of the moment when that beneficial mutation occurs. So from here the system has transitioned to this particular state. So now what is going to happen? Now let us take a look back at the chemostat and see what is going to happen. Either this individual is going to increase in frequency, or this individual is going to be lost from the population because of the action of drift. There might be a chance event that this individual couldn't replicate and was washed away from the system, and hence green individuals are lost. So now this can be represented, the fate of this mutation can be represented as two alternate ways. In one case, the mutation is lost from the system, in which case this graph just becomes same as what we started with, that the mutation was lost and now I am back to a state where there are n individuals at fitness f naught. So this is the scenario where mutation lost from system because of drift. And actually, as we saw in the last video, that the most common fate of a beneficial mutation is going to be this, that it is going to be lost by the action of drift, and it is not going to be able to increase in frequency because of that. On the other hand, if the mutation is able to survive the action of drift, then what is going to happen is that as we move forward in time, there are more number of green individuals, and now the chance that all these green individuals are going to be washed away from the reactor, from the chemostat is extremely small. So, the green mutation is not going to get lost because of action of drift. So, the relative role of drift in dictating the fate of the green mutation decreases as the number of individuals carrying that mutation increases. Now, there are sufficiently large number of green individuals here that drift is no longer going to be the significant player, it is going to be selection which is going to dictate the fate. As a result of that, in this scenario, the frequency associated with F naught plus S will increase, because selection is acting and the more fit individuals are going to get selected for and hence increase in frequency, and conversely the less fit individuals are going to decrease in frequency. So, this relative change will take place once the mutation has survived the action of drift. So, in this graph I have F naught and F naught plus S, N is somewhere here, and what this graph will look like at this stage now is that this would have reduced a lot from N and this would have increased a lot from its starting point, but this is still coming down and this is still going up. So, this relative change in frequency is taking place, and this is at some time point t after the mutation was able to survive drift. Remember that this is the more common fate of a beneficial mutation. And if we let sufficient time pass, then what is going to happen is that the F naught genotype, the ancestral genotype, is going to get completely eliminated from the population, and F naught S will be the only genotype that survives in the chemostat. So, the future fate of this chemostat might look something like this, that there are only these green individuals left in the chemostat. That is because the green individuals, were being of higher fitness, were able to completely eliminate the ancestral genotype. This fate is going to be represented by the following change in the graph, that I have F naught and F naught plus S. This is fitness and this is numbers, and now there is 0 individuals which are at F naught and this is N, and there are N individuals which are at fitness F naught plus S. And this has represented a cycle where a mutation occurred, a beneficial mutation occurred. So, this was the, let me use a different color, this was the event that a beneficial mutation occurred. This was the case that the same beneficial mutation, one, survived drift, and also and thereafter increased in frequency. So, this was step 1, step 2. Alternatively, we can go from step 1 to here, which is the more common fate, but this just brings us back to this particular point and we start over again. And the third step is that as a continued increase in frequency of the beneficial mutation, the beneficial mutation reaches fixation. And it is not as if the process of evolution halts at this point, this process continues as we move forward, and now the population, in the population we are waiting for an event like this to take place, that this happens now, where everyone, all the green individuals in the population are at fitness F naught plus S, and now we are waiting for a event to take place where a newer individual comes into being whose fitness is F naught plus 2S. So, the newer individual is going to be here, this is F naught plus 2S, this has to be changed then, this frequency of the ancestral genotype reduces slightly from n and it comes to n minus 1, and the frequency of the new genotype, the new beneficial genotype that has come in the population is equal to 1. So, in some sense the same process keeps on repeating itself over and over again. This is one view of how evolutionary change could be taking place. One thing that we want to note is that there are two time constants associated with it. So, let me just on a fresh slide quickly sketch these again. So, I started with this and this changes to a new mutation. The beneficial mutation starts to increase in frequency and then it reaches fixation. See, these are the 4 steps. So, this is occurrence of beneficial mutation, this is surviving drift, and increasing in frequency, and lastly this is reaching fixation. And in the case in case that this mutation does not survive drift, then you go back here and this was mutation lost because of drift. And this is how change will take place. Now, we want to understand the 2 time constants which are associated with this particular process. The first time constant in all of these graphs, obviously this x axis is fitness and y axis is simply number of individuals. So, the first time constant is this one, which is the time associated with occurrence of a beneficial mutation which survives drift. Let us call this time as tau occurrence. So, this is the time that I have to wait on an average for a beneficial mutation to occur and it be able to survive drift. Because remember that one is, there are two for this event to happen, 2 things need to happen. One is that there is waiting time associated for a beneficial mutation to occur in the population, and second is that beneficial mutation will more often than not be lost because of drift, and hence after occurrence I have to wait for this beneficial mutation to survive drift. So, there are even within this there are 2 things that we are looking at. First is occurrence of a beneficial mutation, and secondly that beneficial mutation has to survive the action of drift. These 2 things need to happen, because more often than not beneficial mutation will occur and I go back to this state because it is lost due to drift. It will occur again and I will go back because it is lost due to. Last time we saw when the numbers we were doing that actually with reasonable values of S that are found in experiments, 20 beneficial mutations need to happen for one of them to survive the action of drift. So, this tau occurrence represents the combined waiting time associated with happening of the mutation and then its surviving drift. The second time constant associated with this process is what happens thereafter, which is time it takes for a mutation, not just any mutation, but a mutation which has survived drift, to reach fixation. Let us call this time as tau fix. So, these are the two time constants that we have in the process, time of occurrence and surviving drift, and thereafter once it has survived drift, the time to reach fixation. Now, in the picture that we have sort of painted here, we have made an assumption that tau occurrence is much longer than tau fixation, and we will see how that is true. So, let us discuss the first case, that tau occurrence is much bigger than tau fix. What does this regime actually mean? What this means is that if I start with a population and the population is at f naught, n individuals, then because tau occurrence is much greater than tau fixation, tau occurrence can also be thought of as tau wait, the waiting time associated for a beneficial mutation to occur and survive drift. So, this can also be called the waiting time associated with a beneficial mutation to occur and survive drift. How long do I have to wait for these processes to occur? So, in the regime that we are discussing here, tau occurrence is much bigger as compared to tau fixation. What that means is that I have to wait a really long time before a beneficial mutation can occur which is able to survive drift. So, I am waiting, and then eventually what will happen is that this beneficial mutation will occur, and as a result the picture looks like this. This is n minus 1 individuals at f naught and 1 individual at f naught plus s. And then because by definition this mutation has been able to survive drift, the numbers of the ancestral genotype will start to go down, and the number of the mutant genotype will start to go up. So, this change will begin to happen, and the time it takes for this to reach the following state f naught plus s, that time is tau fix. This time is tau occurrence. So, in the first regime what we are saying that tau occurrence is much bigger and tau fix is much smaller. So, as a result, if you imagine this population, the fate of this population in a chemostat is going to be the following: that you have an isogenic population, all individuals are of the same genotype. Eventually, one of them will acquire a beneficial mutation, and this beneficial mutation will survive drift. But that has taken a long time, because that is the meaning of tau occurrence being very high. Once this new beneficial mutation has occurred, tau fix is relatively small. As a result, this new beneficial mutation will go towards fixation really fast, and we will reach this third stage where fixation has happened fairly quickly. After this, we are back to square one, because now we are waiting for the next beneficial mutation, waiting for next beneficial mutation. which is going to be dictated by tau weight, but we know that tau weight is actually very long. So, again I have to wait a long time for this beneficial mutation to take place. So, there are these two processes playing side by side in this process of fixation of mutations. One is occurrence and the other is reaching fixation once occurrence has happened. So, in this particular regime, occurrence takes longer, and hence the picture that we are painting is like this. Now, the question I want to end this video is the following, that if I am operating in a regime like this, so these cells are growing and this is the regime that I am operating in, tau occurrence is much more than tau fix. Then if I look at the system, at an instant of time, how many genotypes do I see? That is the question. And what you should realize at this moment is that the answer to this under this regime should be 1, because in this regime tau occurrence is really long. So, you have to wait a long time for this mutation for a beneficial mutation to occur and survive drift. That means most of the time the system is spending in this particular state where we are just waiting for this mutation to come and system to reach this stage. Most of the time the system is spending is in this particular state of just waiting. Once the wait is over and I reach this particular state, then the transition from here to here is fairly rapid because tau fix is much smaller. As a result, this transition is much fast, and suppose, just to give it sort of made up numbers, maybe I was waiting 10 hours for this beneficial mutation to arise and survive drift, then maybe this fixation only happened in 15 minutes. So, if I take a snapshot of this system at a particular time, chances are that the system will be stuck in this state because this is fairly rapid. As a result, when I take a snapshot of this chemostat, I will only see one genotype at a time more often than not. Occasionally it is possible that I will see two genotypes while this is going to fixation and that one is going to extinction. In that case the answer will be 2 for very few instances of time, but the most frequent answer that you will see in this regime is only 1. And we will continue this discussion in the next video.